A type of virus infecting certain bacteria was first discovered in the early 1900s by an English bacteriologist Watt and was named as bacteriophages by D. Harrell. Soon, different kinds of bacteriophages came into existence, and one among them is the 5X174, our lead actor for today. 5X174 is an icosahedral single-stranded DNA virus infecting E. coli. Close structure of the genome was already discovered. Surprise, surprise, it was the first DNA-based whole genome to be ever sequenced. This work was accomplished by Sanger and his colleagues in 1977, soon after he won a Nobel Prize for being the first person to sequence an entire protein, which was insulin. The genome sequencing of virus revealed important features like initiation and termination sites of transcription and translation, and also the presence of about 5,375 nucleotides with 9 known genes in order of their discovery. Gene A, coded for the largest protein of the virus, required for double-stranded DNA replication and single-stranded DNA synthesis. Gene B, C, and D was required for viral single-stranded DNA synthesis or for DNA packaging, although the function was unclear. Gene E is responsible for host cell lysis. Gene F, G, and H coded for the structural proteins of the viral capsid. The H protein also functions as a pilot protein which transfers viral genome into the bacterial host. Gene J codes for the smallest protein of the virus, which is required for synthesis of basic viral proteins. It was found that the virus codes for proteins with amino acids exceeding the number of codons present in it. It was later established that the proteins could not be separately coded on the DNA. This led to the discovery of an important feature of the virus, which is overlapping genes. Previous efforts on sequencing the virus has led to the sequencing of only a small number of nucleotides, especially in the F and G genes. Sanger developed the plus and minus method, which involved prime synthesis of restriction fragments created by restriction enzymes in the presence of DNA polymerase and radioactive labeled dNTPs by using both the plus and minus strands of the virus as templates. The minus system used only 3 dNTPs while the plus system used 1. Results of both these systems are analyzed by further methodologies for identifying the bases. With the help of previous studies on promoter sequences and the determination of 5' end sequences of the virus mRNA, the transcription start sites were identified in gene A, B, and D at the respective positions. These mRNA start sites have preceding sequences similar to that of a known promoter. Now, the protein initiation and termination sites could be easily located if their amino acid sequences were known. But this is not the case with proteins A, B, and C. Therefore, a different method was adapted where ribosome binding sites were looked for before a particular initiation codon, which could form base pairings at the 3' end of 16S rRNA. In this way, the initiation codon for protein A was located at position 3973 and termination at 133. Another protein, probably a variant of A with lower molecular weight, was also formed by an internal translation site in gene A. It was found that the coding sequence of protein B lies completely within gene A and that the protein initiation codon of gene C overlaps with the termination codon of gene A. The amino acid sequences of proteins D, E, F, G, H, and J are known. By correlating them with their DNA sequences, the results can be estimated. It was seen that the termination codon of gene D overlaps with the initiation codon of gene J, and the coding sequence of gene E lies completely within gene D. The protein initiation and termination sites of gene F, G, and H are as follows. Thus, there are 2 prominent overlappings in the genome of gene B within A and gene E within D. Another interesting feature of the virus's codon is the higher thymine content. This particular study was open to further modifications, and now there are about 5,386 nucleotides with 11 known genes including K in the genome. About 100 more whole genome sequencing of viruses and bacteria was carried out after Sanger. One such example is that of the bacteria Haemophilus influenzae. These projects have thus had a massive impact on improving diagnostic procedures for treating diseases and has opened pathways for new age research in the field of agriculture, infectious disease, and even biowarfare.